Okay, guys, so this is the last FRQ that you have. It's on Unit 6. And remember that Unit 6 is on transcription and translation, protein synthesis. It's also on DNA replication and operons. And so remembering how bacteria are able to regulate their gene expression. And also there was a section that we had on eukaryotic gene expression, looking at ways that we are able to have enhancer sequences that make it more likely for us to transcribe, or silencer sequences and all that good stuff. So this is a question 1. So on your exam, you're going to have question 1 like this. It is a format of experimental design. And so a lot of it is just understanding the components of an experiment, and then also being able to tie in the concept that is being addressed. So in this one, we are addressing the concept of protein synthesis, and they're looking specifically at the enzyme RNA polymerase. Remember, RNA polymerase is the enzyme that is able to take DNA and convert it into messenger RNA in the nucleus, and then that messenger RNA will be able to exit through a nuclear pore to be translated at a ribosome. So I'm going to pull up the protein synthesis notes that you guys hopefully still have. If not, I did post them on Canvas under all those doodle notes and sketch notes that I put under the modules. So if we look here, first thing we're going to have is DNA. Remember, DNA is enclosed in the nucleus. It's the master instructions that we do not want to, like have them degraded by enzymes in the cytoplasm. So we want to make sure they stay protected. And so it's going to stay within the nucleus, but to obtain the instructions and deliver them to the ribosome, we have messenger RNA, which is going to be the one that actually moves the instructions from the DNA to the ribosome to actually turn into the protein. So remember, we have the central dogma of biology is DNA to messenger RNA to protein. And that protein is formed at a ribosome with the help of transfer RNA, which actually delivers the amino acid. Remember, amino acids are the building blocks of proteins, and so you're going to have all of these amino acids are going to link up, and they form a polypeptide chain, and then when it folds and it's functional, it is a protein. So first thing we're going to have is RNA polymerase come in as an enzyme, and it's going to bind at the promoter. We call that the initiation. So first stage is an initiation. It's going to bind to the promoter sequence, and then it's going to start creating messenger RNA. When it creates messenger RNA, we call that elongation. And then eventually it's going to hit a sequence that says, okay, you're done with this set of instructions, and it's going to be the termination sequence. After it reaches the termination sequence, remember our messenger RNA needs to be edited. So we're going to keep the exons. Exons are expressed, and we're going to remove the introns. I relate this to, like, okay, you wrote your rough draft essay, but it's got a lot of like wordiness and you need to cut down on it. And so that would be when you remove the introns, it's just kind of removing some of the fluff that the ribosome doesn't need. Sorry, I had a visitor. So we're going to take our exons, and then we're going to, this messenger RNA is going to exit through a nuclear pore. They're going to attach a 5' cap and a poly A tail so that it doesn't get degraded in the cytoplasm and it doesn't break down this messenger RNA that contains important instructions from the DNA. And then it's going to find a ribosome here, and the ribosome is going to read the messenger RNA, and every 3 bases, we call those codons. Addie, do you want to say hi to my friends? No. Nope. Okay, well, you got to leave mommy alone, okay? Okay. Every 3 bases, the messenger RNA is going to be read by the ribosome, and then a transfer RNA is going to use that sequence to deliver the appropriate amino acid. Remember, the transfer RNA has a little spot on it that will specifically bind to the messenger RNA, and when it says, okay, we matched up, we know that we're delivering the correct amino acid, and then it'll drop off its amino acid as the next one in the sequence. And then we form this long polypeptide chain, which can fold to form a functional protein. If we change any of the amino acids, it's going to change the way it folds, change the shape, change the function. So let's look at this. Scientists studying transcription in yeast created an experimental strain that produced a modified RNA polymerase containing a single amino acid substitution. So my mind goes 2 places here. first of all, RNA polymerase is an enzyme that's, I just want to think, okay, what is RNA polymerase involved in? Okay, it's involved in transcription, the change or the creation of messenger RNA from our DNA template. So right here, this is involved in transcription, creating our messenger RNA template. And they told us that this new modified version has a single amino acid substitution. Remember, if it ends in ASE, that means that it is an enzyme, and an enzyme is a type of protein. And if a protein has an amino acid substitution, that could result in changing the structure, which changes the function. So my mind automatically right here, the single amino acid substitution, I'm thinking, okay, if we're changing one of the amino acids, then we're changing the primary structure of the protein. If we change the primary structure, it can change the whole folding pattern. And if we change the folding pattern, we're changing the shape. And if we change the shape, we change the function. So could this affect how well RNA polymerase functions at converting DNA to RNA? So that's kind of where my brain goes, just reading that. The scientists determined the maximum elongation rate during transcription with and without the modified RNA polymerase. So we want to see, now that we've modified this RNA polymerase, how effective is it at converting DNA to messenger RNA? And remember, we have initiation where it binds to the promoter region, elongation where it actually creates the messenger RNA, and termination where it stops. So the maximum rate at which it's creating messenger RNA is the elongation. So if we look right here, we have our wild strain and our experimental strain. This is the one with the modified RNA polymerase, and we're looking at the maximum elongation rate. Our independent variable is which strain we're using. Our dependent variable is the maximum elongation rate and the nucleotides per second. And so how fast are they making messenger RNA based on whether or not you have the wild type, meaning the naturally occurring RNA polymerase, or the modified RNA polymerase that contained a single amino acid substitution. When I look at this right away, I see that the elongation rate or the creation of messenger RNA is way higher in the wild strain than it is in the experimental strain. I can also look and see my error bars are not overlapping, so I know that there is a significant difference between the rate of elongation between these two. And I can say that this data would be slightly less reliable because it contains a larger error bar. So remember, those are what we can learn from error bars, and go ahead and anticipate any questions that they could ask you. I haven't even read what's going on in figure 2, but I can see that their error bars would overlap, so there'd be not a significant difference between these two. Okay, the compound amanitin, I don't know, I can't read, which is commonly found in toxic mushrooms, is a specific RNA polymerase inhibitor. So remember, RNA polymerase is an enzyme. An inhibitor would do something to reduce the effectiveness of the enzyme. So a competitive inhibitor would bind to the active site, and a non-competitive inhibitor would bind to an allosteric site. Both prevent the enzyme from breaking down its substrate or completing its task. That amanitin binds to the RNA polymerase active site, so it's a competitive inhibitor, and inhibits transcription. In a second experiment, the scientists treated the wild type and experimental strains of with a 40 microgram per milliliter solution of the amanitin and recorded the maximum elongation rate of the mRNA. Error bars represented 2 standard errors. So right here we can see the difference between the wild strain and the experimental strain. With the amanitin, there is not a significant difference between the rate of elongation. Here, the rate of elongation of the experimental strain is still low because the change in the amino acid substitution clearly reduced the effectiveness of the RNA polymerase. And then here, with the RNA polymerase being inhibited, it's not working well, and we don't have a very large elongation rate either. So both are affecting the rate of elongation. All right, let's just get into our questions now. Describe the 3 structural components of RNA nucleotide monomer. I thought it was kind of a weird question. It was such like a content question. But the 3 portions of any nucleotide are a sugar, phosphate, and a base. Remember, in DNA, the sugar is deoxyribose. And then our bases are the nitrogenous bases, adenine, thymine, cytosine, and guanine. That's for DNA. But here, an RNA nucleotide is going to have a sugar, a phosphate, and a base. The sugar is a ribose sugar because it's ribonucleic acid. The phosphate is still phosphate. And then the bases are adenine, uracil, cytosine, and guanine. So sugar, phosphate, and a base. And if you want to articulate that the bases are going to be adenine, cytosine, guanine, and uracil. And do specify that the sugar is a ribose sugar. Explain the role of RNA polymerase during transcription. So if you guys remember, RNA polymerase is the enzyme that converts DNA into messenger RNA. So the role is using the DNA template to create a strand of messenger RNA that can be delivered to the ribosome to be converted into a protein. So your role of RNA polymerase, again, is to use the DNA template and convert it to messenger RNA. And if you want to elaborate that the messenger RNA can then travel to a ribosome to create a protein, that would also be good information to add. Identify the dependent variable in the experiments. So they said both experiments, and if we look, our dependent variable is the same either way. So you don't have to specify two different dependent variables, it's just one. Remember, dependent is always on the y-axis, and here our dependent variable is the maximum elongation rate. Identify a control group missing from the second experiment. So justify the need for this control in the second experiment. So here we have the wild strain is treated with the amanitin. The experimental strain is also treated with the amanitin. And we would like to identify a control group that we should have. So typically your control group is just the one that is not treated with something. So it does not receive your independent variable. So here our independent variable is whether kind of like they're both receiving this amanitin. And so we would want a control group that does not receive the amanitin. So we would use either, you could use both, the wild strain without the amanitin and compare it, and the experimental strain without the amanitin and compare it. And just the need for that is to show that the amanitin is reducing the elongation rate, to show that there is an impact of using the inhibitor that is actually causing the elongation rate to be reduced. So if we just had the experiment on the right, we wouldn't know if the amanitin was actually causing the elongation rate to be lower. We'd need to compare it to experiment number one. So a control group that you would have is either the wild strain or the experimental strain without being treated with the amanitin, so that you can know that the amanitin actually is affecting the rate of elongation. So the identify the control group is the wild strain or the experimental strain or both without being treated with the amanitin. And the need is so that you can prove that it was needed or that you can prove that it was the reason that the elongation rate has been reduced. Describe the effect of amanitin on the maximum elongation rate for the wild type and modified RNA polymerases. So for both the amanitin, in figure 2, for both the wild strain and the experimental strain, our axes are the same. That's important to check and make sure that your numbers are the same on the side. But we can look here, our elongation rate, the average was like 12. And over here, it's like 1. So here, this maximum elongation rate average was about 2. And here the elongation rate was like, well, this is probably even less than 1, honestly, when I look at it, because I would assume halfway here would be 2. So, I don't know. I guess this is closer to, this is like closer to like 0.5. But basically, you can see that it reduced the elongation rate for both the wild strain and the experimental strain. Determine the ratio of the average maximum elongation rate for the modified RNA polymerase compared to the wild strain RNA polymerase in figure 1. So we want to figure out the ratio of the average maximum elongation rate for the modified RNA polymerase compared to the wild strain RNA polymerase in figure 1. So we want to look at the rate of both and then create a ratio. So up here, this was the elongation rate average is where the bar peaks, not the error bar. That shows us the range of the data, but this is the actual mean of the data is 12. The rate is 12. And then here, again, you can tell this one's about right in the middle. It's a less confusing about what number they're trying to have us identify. And I would definitely say that was 2. So our elongation rate for wild strain versus experimental strain would be 12 to 2, and we could reduce that down. And I'll let you guys do that really difficult math on your own. But you would have the rate of 12 in the wild strain nucleotides per second versus 2 nucleotides per second in the experimental strain. And again, you would need to reduce that ratio down to get the correct answer. But I'm going to let you guys reduce that fraction on your own. Okay. State the null hypothesis for the experiment in figure 1. Provide reasoning to justify the claim that the change in the amino acid sequence in the modified RNA polymerase affected the shape of the active site on the enzyme. All right, so if you guys remember, a null hypothesis states that the independent variable has no effect on the dependent variable. So first, when I read that one, I had to go back and say, what was being tested in figure 1? Well, figure 1 is just looking at the elongation rate of the wild strain versus the experimental strain. So the difference here, what is the difference between these two? Well, the difference is that this one in the experimental strain has modified RNA polymerase. And so the null hypothesis would say that the modified RNA polymerase will have no effect on the rate of elongation. So remember, it says that the independent variable, or what is being tested and compared, will have no effect on the dependent variable. So the difference between the two on the x-axis will have no impact on what we read out on the y-axis. So the modified RNA polymerase, or the single amino acid substitution in the RNA polymerase, will have no impact on the maximum elongation rate, would be our null hypothesis. And again, remember, a null hypothesis says that the independent variable, what you're changing, is going to have no impact on what you're measuring. So if we see a difference between the two, especially if their error bars do not overlap and there's a significant difference, we can actually start to say, okay, the independent variable, the change in the amino acid sequence of the RNA polymerase actually did impact the maximum rate of elongation, because you can compare the two and see their error bars not overlapping and that there must be a difference. And so they don't even ask you to analyze the error bars here, but that could be something you would have on your actual exam. So null hypothesis is that the modified RNA polymerase would not have an impact on the maximum rate of elongation. Provide reasoning to justify the claim that the change in the amino acid sequence in the modified RNA polymerase affected the shape of the active site on the enzyme. Well, the evidence would be that the experimental strain had a much lower rate of elongation. You could bust out the error bars here if you want to talk about that. It was a significant difference between the two. And that the changing in the amino acid sequence would change the primary structure of the protein, which would change the way it would fold, so that the active site would have a different shape and potentially would not be as effective at converting the DNA into messenger RNA, because of the different shape would cause it to have a different function. And so the reasoning would be that the amino acid substitution would change the primary structure, which changes the way it folds, which would change the active site of the enzyme. And you could also use the evidence up here to say that the graph shows you that there was a significant difference between the two strains when the active site was changed due to the amino acid substitution changing the primary structure. So I hope that makes sense, and I hope that helps. And good luck with all your studying. Please tell me any ways that I can help you. If any topics you want me to talk about, review, happy to help. We could do Zoom conferences the week leading up to the exam if you just want to talk over the notes and strategize ways to study. But just kind of let me know what you guys need from me, and I'm happy to help. So have a great day, and bye-bye.